So where do antibodies come from? They come from B cells. And so now I'm going to talk about B cell sensitization and activation. So in step 1 here is the inactive B cell, and it displays antibodies for a specific antigen on its surface, as well as class 2 major histocompatibility complex on its surface as well. And so here's the antigens that are going to sensitize this B cell. So the antigens are recognized by the antibodies, and that causes the B cell to be sensitized. In order for the B cell to be activated and to begin to differentiate into a cell that can create antibodies, it has to be helped by the helper T cell, which, remember, is that CD4 T cell from the last lecture. The helper T cell has its T cell receptor that recognizes the antigen, along with the MHC class 2 molecule that was on the B cell. So it's the interaction between those two, and then of course you have the CD4 that's not shown here, that leads to the activation of that B cell. So the sensitized B cell becomes active. This also requires stimulation by co-stimulation by cytokines like interleukins. And this stimulated B cell is then going to proliferate into many different activated B cells, as well as memory B cells, which hang around for the next time you see the same infection. But eventually these cells are going to differentiate into these large plasma cells, which will start producing antibodies to the antigen. Almost 100 million antibodies are produced per hour. They make a lot of antibodies. But the first time that a B cell gets activated and gets sensitized, it can take a long time, 3 to 14 days, for the antibodies to actually build up enough in the blood to have an impact. And so for this reason, the first time you see a specific pathogen, you may be sick for a couple weeks before your body can actually fight it off. But if you see that same pathogen again, those memory cells are going to pick right up, and you'll start creating antibodies to that antigen right away during the secondary response.